Hello everyone, my name is Michael Charles and today I will be talking with you about the biology and history of the greater chestnut weevil Curculio caryatropes. It is a host specialist on American chestnut. As I am sure we all know, there are over 400,000 species of beetles and over 60,000 species of weevils. So, before I got started, I wanted to take the time to point out where this genus is located phylogenetically. Curculio is located down at the bottom of this phylogeny, indicated by the red arrow we see here. There are estimated to be about 500 species of Curculio, and they are distributed worldwide except for in Antarctica and in South America. After being oviposited, the eggs would typically hatch within about 5 to 10 days. In this picture on the right, we have some eggs and we can see that they are creamy colored and slightly opaque. After hatching, the larva would feed on the nut for about 20 to 30 days. When it was finished feeding, the larva would chew its way out of the nut, which we can see here in this image on the left, and it would burrow into the soil. This image on the right gives us an idea of just how much damage these weevils can cause to the nut. We see all of the cavities, the rotting parts of the nut, and just the pathways that they have made whilst feeding on the nut. The larvae would burrow into the soil to a depth of up to about 12 inches, at which point they would form a smooth-walled cell in which they would eventually pupate. In this image on the right, we have a Curculio pupa, and we see it within its smooth-walled cell in the soil. After pupating, the adults would remain in the soil for an additional several days to allow sclerotization to occur. In this image on the right, we have a male specimen of Curculio caryatropes traversing on the burr of an American chestnut tree. The adults would emerge from the soil in between the months of July and September, typically peaking in the month of August, at which point they would make their way to the catkins and burrs on the American chestnut trees that they would find above them after emerging. In this image, we have 4 specimens of Curculio caryatropes. We have 2 females, these 2 here with the longer rostrums, as well as 2 males here on the sides, here and here, with the shorter rostrums. After making their way to the burrs and catkins above, mating would typically occur between the months of August and October. In this image, we see 2 specimens, a male on top and a female below, mating on the burr of an American chestnut. We can even see the ediogus of the male here. After mating, oviposition would also typically occur between the months of August and October. Females would use their long rostrum to bore into a developing nut. Afterward, they would align their substitutional ovipositor with the hole that they just made and deposit an egg into the nut. In this top image, we have a specimen of Curculio with her rostrum almost fully inserted into the nut as she is boring into it in preparation for depositing an egg. In the bottom image, we have this same specimen turned around with her ovipositor placed above the hole, depositing an egg. We can see the substitutional ovipositor as well as part of the egg being deposited into the hole itself. The American chestnut, Castanea dentata, had a natural range that stretched from Maine to as far south as Florida, and from the east coast to as far west as Illinois and Mississippi, which we can see here in this map on the right. When flowering, castanea are rather easy to identify by the presence of long white catkins, these structures here, and green spiny burrs, these structures here. The American chestnut is also the only documented host for Curculio caryatropes. Unknowingly imported to the United States on Asiatic species of castanea, the chestnut blight, Cryphonectria parasitica, was first noticed on an American chestnut tree at the New York Zoological Park in the year 1904. This necrotrophic fungus enters through cracks in the bark of the tree and causes cankers to form, which then interfere with the tree's ability to transport water and other nutrients from the roots throughout the rest of the tree. This will essentially girdle the tree and eventually results in its death. The root system, however, remains intact as it is not affected by the blight and allows for new sprouts to grow. Unfortunately, the sprouts will still be susceptible to blight and most will die before reaching reproductive maturity. Because the greater chestnut weevil is dependent on the nuts for food, the functional extinction of the American chestnut has led to the drastic decline and likely extinction of this weevil. We see here in this image on the right the canker that is formed by this blight. And although this will girdle the tree, the roots are not affected and allows new sprouts like these in this image to grow from the original root system. One aspect of my research involves documenting the decline of the greater chestnut weevil through time. To do this, I have reached out to numerous entomological collections and received the label data and locality data for 294 specimens of the greater chestnut weevil. Of these 294, unfortunately only 184 of them have date labels. This figure from Gravatt, dated 1949, illustrates the spread of chestnut blight throughout the natural range of the American chestnut. The key shows that the dot-dash line is the natural range of the American chestnut. The dashed line indicates the limits at which 50% to 99% of the chestnut trees have succumbed to the blight, and the shaded area indicates the remaining area in which the trees are mostly infected. We see the introduction of the blight introduced up here in New York City in 1904. By 1910, the blight had spread to the adjacent states. By 1920, we see the blight had reached Maryland. By 1930, the blight had spread throughout about half of the natural range of the American chestnut. And by 1940, it had devastated populations as far west as Indiana and as far south as Georgia, and it had infected trees throughout the remaining portion of the southern native range. Over here, we have the number of dated collection records by decade for the greater chestnut weevil. In the 1880s and 1890s, there were 11 records total. In the 1900s, there were 56 records. In the 1910s, there were 57 records. In the 1920s, there were 39 records. In the 30s, there were 3. In the 40s and 50s, there were 7 each. There were none in the 60s and 70s, and there were 3 in the 80s, with just 1 in the 1990s. We can see that the decline in the collection records of the greater chestnut weevil appeared to coincide with the increased spread of the chestnut blight throughout the range of the American chestnut. Unfortunately, as there have been no collection records for this weevil since August of 1997, it would seem likely that the greater chestnut weevil is extinct. I would like to thank the University of Memphis Center for Biological Diversity, the Harvard Museum of Comparative Zoology, the Smithsonian National Museum of Natural History, the McKenna Lab, Dr. Lourdes Chamorro, Dr. Robert Anderson, and numerous entomological collections for their support in my research. And with that, I would like to take any questions that you may have.